Good afternoon, everyone. I'm Ken Luchin, Dean of the College of Engineering. I welcome you to the Charles DeLisi Distinguished Lecture event. We have two wonderful awards to give out today. One, of course, will be to Joyce, which I'll introduce in a few minutes. But first, I will give out the award for the College of Engineering Early Excellence in Research Award. This year's winner will be Mo Khalil. Let me tell you a little bit about Mo. Mo got his bachelor's in 2002 in mechanical engineering with a chemistry minor from Stanford University, his master's in mechanical engineering from MIT, and his PhD in mechanical and biological engineering also at MIT, where he worked with Angie Belcher, who has been our graduation speaker, by the way. In approximately 4 or so years as a BME faculty member, Mo has become internationally recognized in the now, and in fact, maybe an emergent leader in the field. It used to be brand new, but now it's not so new anymore, in synthetic biology, and specifically the frameworks for engineering eukaryotic or higher-order organisms. He's currently the Associate Director and founding member of the new Biological Design Center. In fact, within just a few years, about 3 or 4 years after beginning his faculty position, he's published multiple papers in some of the most competitive, high-quality journals in the nation, including, for example, a paper in Cell, which was chosen as the journal cover and highlighted by Nature Reviews, Genetics, and various media sources. He's been asked to write reviews on emerging topics in Nature Reviews, Genetics, and Genome Biology, and a perspective piece in the journal Science. He's published over 30 publications in peer-reviewed journals that have been cited over 2,400 times. And he has an astonishing record of funding success, including working backward from old days to now, a Genome Web Young Investigator Award, an NSF Career Award, a National Academy of Engineering Frontiers of Engineering Education invitee, a Hartwell Foundation Individual Biomedical Engineering Research Award, a DARPA Young Faculty Award. He is a PK, so a Presidential Early Career Award for Science and Engineering Award winner, and an NIH Innovation Award. If you need money, you go to Mo. And there's more, which are my favorite awards. In 2015 and 16, he got the award for teaching excellence in both those years in biomedical engineering, and in 2016, the Outstanding Professor of the Year for the entire College of Engineering. Please join me in congratulating Mo Khalil as the College of Engineering Early Excellence in Research Award winner for 2017. Mo, stand up. Come on up, I'll shake your hand. It's supposed to be a check, but we ran out of money. He's got plenty of money. Now let's transition to the event itself, the College of Engineering Charles DeLisi Distinguished Lecturer. This year is Joyce Wong. But first, some background. This annual Charles DeLisi Distinguished Lecture celebrates the career and research impact of a senior faculty member or an alumni that was mentored by a faculty member of the College of Engineering. Each year we solicit nominations from the entire faculty, and a review panel of luminary faculty, including many of the previous award winners, recommend a finalist. Now, the lecture series is made possible by a generous gift that bears his name from one of the most historically important figures in the history of the College of Engineering, Dean Emeritus Charles DeLisi. Charles arrived at Boston University as dean around 1990, when we as a faculty were contemplating how to become a world-class research college of engineering. It turns out, fortunately, we didn't have to contemplate anything. Charles knew exactly what we needed to do to propel ourselves forward by recruiting an extraordinary set of junior and senior faculty members, who then attracted internal and external resources, including other great faculty, that allowed the college to spin its magic. In just 1 decade, we were on the map to stay as a world-class College of Engineering from a research point of view. He, in addition to being an incredibly high-impact dean, however, Charles is far more than that. He's literally considered one of the most crucial national figures in the history of the Human Genome Project, and the only person to receive, so far as I know, the President's Citizens Medal from President Clinton for essentially being the father of the Human Genome Project. He is a visionary, as well as a great scientist, as well as a great leader. And I ask us all, I ask Charles to stand up, and all of us to give him a hand to thank him for the impact on the college and this gift for creating this wonderful distinguished lecture. Thank you, Charles. This year's lecturer is Professor Joyce Wong. She is the Distinguished Professor of Engineering in the Department of Biomedical Engineering, the Division of Material Science and Engineering, and she also has an appointment in General Medical Sciences. She's a member of the BMC Cancer Center. She's the director of ARROWS, which stands for Advanced Recruit, Retain, and Organize Women in STEM at Boston University, out of the Office of the Provost. She is co-director of the Affinity Research Collaborative in Nanotheranostics. I don't know what that means. She's a member of the Whittaker Cardiovascular Institute. She's a member of the Center for Regenerative Medicine, a member of the Center for, or the BU Nano Center right now. It used to have a former name. She's a faculty of the Biomolecular Pharmacology program and the Molecular Biology, Cell Biology, and Chemistry program. She's a member of virtually every program in the university that is related to biology and science, and deservedly so. Now, she received her bachelor's in material science and engineering from MIT, and she received her PhD program in polymer science and technology in the Department of Material Science and Engineering, also from MIT, where I believe she worked with Bob Langer. In 1994, Joyce was a postdoctoral fellow in biophysics at Surface Science at the University of California in Santa Barbara. And in 1995, was a visiting scholar at the University of Mainz in Germany. In 1998, she has been on our faculty at Boston University. She served as the Associate Chair for Graduate Studies in Biomedical Engineering, as well as the Associate Director for the Center for Nanoscience and Nanobiotechnology, its former name. Her research focuses in areas of biomaterials that detect and treat disease, with her current focus to elucidate fundamental molecular cellular and colloidal interfacial processes and apply these to pediatric vascular tissue engineering, and to develop in vitro engineering model systems to study disease progression. The specific applications are to detect and treat cardiovascular disease, like atherosclerosis, vulnerable plaques, and the building of vascularized tissue, as well as cancer metastasis. She applies her basic research for enhanced oil recovery with magnetic resonance nanoreporters as well. She is recognized for her service to BU in this institution and externally. In 2015, she was awarded the College of Engineering Faculty Service Award. She is an editorial board member of 6 journals related to biomaterials, tissue engineering, and regenerative medicine. She is a 2017 Materials Research Society Bulletin Volume Organizer, was chair of the Gordon Research Conference in Biomaterials and Tissue Engineering in 2011, served on the National Academy of Engineering's German-American Frontiers of Engineering Organizing Committee, and is conference co-chair of the recent 90th American Chemical Society Colloid and Surface Science Symposium in 2016. She has been selected twice to participate in the prestigious Kavli Foundation Salons, and Dr. Wong is a fellow of the American Institute of Medical and Biological Engineering and a fellow of the Biomedical Engineering Society, and was elected to serve on the BMES Board of Directors. She's received an NSF Career Award, Claire Booth Luth Assistant Professorship, DuPont Young Professorship, Hartwell Individual Biomedical Research Award, and many others. In addition to training 13 postdocs and 25 PhDs and 15 Masters of Science trainees, she dedicates a significant effort to training undergraduate researchers. She's trained 58 senior project students as a faculty member and is a mentor of the BU Beckman Scholar Program. She has over 90 peer-reviewed manuscripts that are published, cited over 6,000 times. 21 have 23 undergraduate co-authors. Many of our undergraduate trainees have gone on to win prestigious awards, such as the Barry Goldwater Scholar Award, or the Forbes list of 30 under 30, or NSF Graduate Research Fellowships. Finally, on a personal note, Joyce was a young faculty member when I became chair of biomedical engineering. No kids yet. Look at the size of these kids she has today. To be so proud, that should be so proud of her. I watched Joyce grow into this incredibly charismatic, caring, fearless, and creative scientist that takes on every challenge needed for a research-driven tenure-track faculty member and brilliantly succeed in teaching and research and service. She's also one of the best networkers I've ever met in my life and one of the world's best people and colleagues. Please join me in congratulating and welcoming Professor Joyce Wong as the 2017 Charles DeLisi Distinguished Lecturer.
Thank you. Thank you, Ken. Thank you for your kind words. And I just want to, actually, I'm really thrilled to be able to give this talk because it gives me a chance to say thank you to so many people here at BU, primarily Charles. I just have to say your vision in creating such a wonderful environment here at BU, starting from as Ken said, all of the people that you recruited here, the environment, making BU on the map with the molecular. It was just so exciting, and I'm just honored to be here, and just thank you for everything. So what I thought I would do first and foremost is I'd like to thank, it's my lab. I think my lab is mostly over here. I wouldn't be able to do anything without them. So one of the themes you'll hear throughout this talk is teamwork, and how I rely, as Ken said, I do a lot of networking, but as you know, science requires a lot of work from many different people. So I have various old lab pictures. This is a retreat we took actually up in Stowe, Vermont, skiing. And this is a more recent picture, but another theme you'll see here is science is also very international. So I have all the flags here of the former and current students in my lab, visiting scientists and things, and there'll be a quiz at the end on all the flags. So just but to be serious, I think it is timely right now for us to keep in mind too that science is global, and we need to kind of keep that in mind. So I also wanted to thank all of my current and past BU collaborators. It was kind of difficult finding all of you online, but I did manage to find some pictures that are on there. But these are basically you'll see they're not only from engineering, but they're from the College of Arts and Sciences. They're also from the medical school and other places. And so these are all people that I've either published with. Some are no longer here, but I'm still very close with them. Some of them co-advise senior projects. So I'm also very grateful for this environment. And I'm also very grateful for the entire BU environment. So the other faculty at BME serving on my students' thesis committees, and also staff, but I noticed none of the staff are on the website. I don't know what the pictures are, so maybe, I don't know if that's something we need to do. And then also, thank you to the colleagues around at BU that really makes it an exciting environment. So what I thought I would do first is tell you a little bit about myself. I grew up in Michigan. Those of you from Michigan know we always show the hand. So I'm from here, like St. Clair Shores. And I'm from the Motor City, and this is important to know because the big three, General Motors, Ford, and Chrysler at the time, you just cannot be in Michigan without being affected somehow by the auto industry. And so in that sense, just to give you a little bit of sense, polymers were in my life from a very young age. This is my mother here. Oh, so here's the flag. So she was born in China, but she was at the Institute of the Polymer Institute at University of Detroit. Just as another theme I'll talk about is the women networks in my life and my career that have been very important. So just as a side note, my mother got both my mother and father got their PhD in physical chemistry at University of Michigan, and so my father had gotten transferred over to Uniroyal in Detroit, and so she was looking for a job. And first she wanted to be a teacher, maybe teaching chemistry at a high school, but they wouldn't let her teach because she didn't have an education degree. So the next thing she did, she just my mother was fearless, and she taught me that. She basically switched fields and decided, okay, I'll learn about polymers. So then she just studied polymers. And then I remember this I found off the internet, but it reminds me a lot of us playing in her lab, she did a lot of polyurethane foams. And actually collaborated with Art Corey, who's sitting here. And so, but my memories of playing in the lab and also helping, English was not her first language, so I helped edit some of her papers and things. So maybe that was kind of getting into my mind about polymers. And then the second thing was when I was in high school, my father worked at Uniroyal. So you notice this is Taiwan. Those of you may know China and Taiwan. That's a whole nother story, right? So they were married from... But anyway, so Uniroyal was a great way for me to learn about composite materials, and I spent the whole summer basically ripping tire cords out of tires and measuring their tensile properties, okay? So that's what keeps you safe, that you have good tires. So my dad worked a lot on tire tread. And so the other thing I did in college, I would come back in the summers and work at General Motors. So those of you may not remember, there's actually the Pontiac Fiero, which was all, outer body is all plastic. I don't know if you know that. It's completely plastic outer body. They don't make it anymore, but that's what I was doing. And so I was, going back to MIT for graduate school, and I thought, okay, I'm going to study, that was in my research interests, I'm going to study high performance materials for making cars, until I went away in the IAP period. Those of you who know MIT, there's an independent activities period in January, and I wanted a little bit of a break from MIT. I had been there, basically 4 years plus a semester. And so I had a long conversation with my uncle here, who's the professor in molecular biology, and we just basically stayed up all night talking about his research, which was on viruses. And so this was in Taiwan. And so when I came back after the IEP, I sought out Bob Langer, as Ken mentioned. So he was fortunate to take, I was fortunate, oh no, don't tell him I said that. That's not what I meant, I was so nervous. Okay. I was fortunate enough to be in his lab, and then so I worked in his lab, and also with Don Ingber. And so I worked with electrically conducting polymers and looking at cell phenotype and basically doing that. And so that was my kind of hint that I was interested in a little bit of biology and trying to integrate that with polymers. So then I did a postdoc, and this is just beautiful. I mean, how can anyone leave that? That's UC Santa Barbara, and it's like that every day. Okay, and that's a true story, because I remember when I first got there and in the lab, I was like, why isn't everybody outside? Why is everyone inside working? And then I realized, because it's like that every day. And then, so then I worked with Jacob Izrielashvili and Joe Zazazinski, the most difficult names to pronounce, and working with liposomes and micelles and model systems. And so when I came to BU, starting my own lab, I first want to acknowledge the mentors. First of all, Charles, I love this picture I found on the internet, because you were so welcoming. So here you are, father of the Human Genome Project, but you would let us come into your office and just talk to you about science, and that was just wonderful. And you had hired Micah Dembo, who hired me, and that was just wonderful working with Micah and learning about cell mechanics. And Ken, you were amazing. I don't know if you remember this. You actually sent me, as an assistant professor, you said, you need to meet some program officers. So I don't know if you remember that. You sent me down, me and a couple other assistant professors down to NSF, just to have, just to meet with them, and that was fantastic and great. Laurel Carney kept saying, when are you going to send your RO1 in? When are you going to send your RO1 in? So she was always kind of bugging me about that. Matt Nugent was a postdoc in the Langer labs when I was there, and it was just wonderful, because at the time he was in biochemistry. So it was great to have him as a mentor as well. And then I put Jim and John on here because I kind of co-opted them. I don't know if you remember this, because I had talked to one of my colleagues over at MIT who was saying they had mentors that they met with, every year, and I said, I want that too. So I just basically said, John and Jim, will you be my mentors? And fortunately they said yes. And so then I also had other mentors. So this is Tosh Chokoti. Some of you may know he's currently chair by BME at Duke University. But he was amazing in terms of helping me and giving advice and things. And then also, I put this picture up here. This is Jean Chin. She was actually my postdoc, NIH NRSA postdoc advisor, not advisor, program manager. And she recently retired. You know you're getting old when your program officer retires. But basically, what she posed a question to me, which I think was really good, is she asked me, as an engineer, do you want to create a better tool to look at, an old problem in a new way? Or do you want to study a particular disease? And I remember her asking me that, and me thinking really, really hard about that. And I thank her for that, for asking me that question early on, because through that I decided I wanted to focus on cardiovascular disease for a number of reasons. My father had suffered, a heart attack not too many years before that, but I really have Jean Chin a lot to thank for that. And then also, as Ken mentioned, this theme that I'll touch upon later too is I was a Claire Booth-Luce assistant professor here at BU, and she was just very inspirational. And so then this was kind of the beginnings of my lab. And so my lab currently, it does 3 areas. I'll basically talk about 2 of them today. The Theranostics, I'll tell you, Ken, it's basically, are you listening? Paying attention? There's a quiz after, remember? So basically, it's a conflation of therapeutics and diagnostics. So it's an easier way to say is to see and treat. And so the first story I just want to tell you basically is vascular patches that grow with a child. And here's a picture I want to show you. Oops, sorry. Whoops. Let me go back. Here, this is Ram Amani, who really is such an inspirational collaborator. And so what he does every day is he basically sees patients and cures children, babies, with congenital heart defects. And so when I was fortunate to be on sabbatical and observe surgeries with him, I saw like the youngest was a day-old baby where they had done surgery in the fetus, right, and actually gone through the valve and had to do surgery as soon as the baby was born, to a 26-year-old who was having like their 6th surgery, right? And so this is kind of the topic we're interested in, is how do you build these patches? When I was watching these surgeries, it was interesting to me that he was basically taking from the pericardium different patches of the heart and then using that to sew on kind of as suture material and just like kind of patch material, like you would patch up your jeans, right? And so when seeing that, how we would do that, it inspired us to develop these patches. And as a side, I want to Ken has mentioned funding, right, which I think is so important. And I just mentioned this too, just as I know Congress is back in session, after the Easter break. And I think it's so important to realize that a lot of this work, whether it's basic science that then becomes, evolved into applied research, that there was many, many different foundations and institutions, the National Science Foundation, National Institutes of Health, and many other institutions that funded this. So this is really important to keep in mind as well. So just to tell you a little bit about the need, there's a very high unmet clinical need. About 8 out of 1,000 infants are born with congenital heart disease. So this here is a normal heart. You have these congenital heart defects. Sometimes you even have where the aortas are actually switched on the different valves, and so I've actually seen that they have to cut them and switch them. So they're basically doing, if you will, kind of replumbing of the heart, and you can see here that you can have a vascular patch that's put here. But the current solutions actually have a lot of problems. So some of them, they use, homographs, so they may take it from the patient, but you see every time you have to go in again for surgery, there's a lot of scarring. So you have to have, it's not ideal, and you have limited tissue that you can draw upon. You have bioprosthetic or prosthetic valves, but a lot of these kinds of things calcify, and it's a real problem. And more importantly, the synthetic grafts, they can't grow with a child, so that's why I said the 26-year-old was on their 6th surgery. So there would be a whiteboard there that would say, okay, this is their X number surgery, and what is the stage of the surgery. So as I said, a lot of times you have to have repeated surgical interventions. So one of the things that my collaborator Ramamani, he was first involved with, in part of, he also has a research lab. And so he was looking at stem cells and trying to differentiate them into multiple cell types, but finding that they would lead to vascular graft calcification. And why this is true is you could have stem cells that can become fat, bone, or cartilage. And so what basically the point is that the cell source is extremely important. You have to think about your cell source. And in thinking about that is how do you actually build a vessel or build a patch? So for that, you need to understand a little bit about these blood vessels. And it harkens back a little bit to my exploration with the tires, right, which are composites, right? There they had the tire cords that were the strength. And here they have collagen fibers, right, that are the strength. So you have these cylindrical kind of layers that it's interesting, they have helical organization of the cells and of the collagen. And so they have this kind of herringbone structure when you look down on it. So you can also look at this and say, I can make a patch out of this by actually patterning them and then layering them into different organizations and then making them into a patch. So this was our strategy. And one thing here to mention too is that the main cells that are in the meat of the vessel are vascular smooth muscle cells. And so just to tell you a little bit about the cell source, this is work that my student Erin Roberts, and this is again, she's co-advised by Ramamani. So Ram had this brilliant idea saying that can we basically harvest cells from placenta? The placenta is normally just thrown away, discarded at birth, but it turns out that they could be a very rich source for stem cells. And the question is can we harvest these and select out and turn them selectively into heart cells and make them into a patch? So that's one way. And another student, George Kwong, who's co-advised by Daryl Cotton over at the Center for Regenerative Medicine at the BU Medical Center. So he's a developmental biologist and a clinician. And so basically, you can use these induced pluripotent stem cells, which some of you may know that the Nobel Prize was given to that a few years ago, where they basically found a small number of transcription factors where you can turn any kind of adult cell, revert it back into a stem cell, and then by adding particular factors, you can control them and becoming them into different cell types. So what George has done, what I'm not showing on here, he's actually developed a couple of reporters that you can see by something called fluorescence, where you can basically, they shine different colors when you excite them with different light wavelengths. And these smooth muscle cells, he's analyzing these right now to see what kinds of vascular smooth muscle cells they are, and then he wants to build these into these sheets that I talked about and measure their functionality. So the question now is, how do you make a sheet of cells, right? And so fortunately, this group in Japan, the Okano lab, we collaborated with them, and so they had actually developed this really cool technique. This, again, polymers is another theme coming back. So this is called a thermoresponsive polymer that's really kind of cool, and it responds to temperature. And so basically, above a certain temperature, the polymer really likes itself, and it basically wants to be kind of in a compact state. And so in that state, the cells can attach to it and spread very nicely. But below that temperature, the polymer likes to be in the solvent, in this case, which is water, something aqueous. So then the cells can then basically come off, and they come off as a sheet of cells. And so how this happened, again, is this theme of this international kind of collaboration, is Yukiko Tsuda was a visiting student from Japan from the Okano lab, and she basically worked together with my postdoc, Brett Eisenberg, and Corinne Williams, a graduate student. And so they had done in the Okano lab basically any organ you can think of, right? So they had done skin, the eye, the kidney, liver, the heart, but they had never done patterning on this. So what we did is we said, can we then merge the two and basically do micropatterning and make sheets of these. And the long story, I'm not showing you all the data, is that you can do that. And here, just to show you a quick movie on this, is you can have basically this sheet of cells. This is just by turning the temperature on. You can actually put like a layer of gelatin to then stack and basically stack these. So that's what Nicholas here has done. He's basically stacked them at different angles, because I told you, remember, the cylinders are at different kinds of angles. And I forgot to mention that the angles depend on where you are in the vasculature and what species it is. So now that we showed that we can basically stack these, we wanted to measure their mechanical properties. And so that's where Dan Backman, one of my PhD students, he developed this very cool mechanical stretcher, and it's based on the Hall effect sensor, so he's using magnets on this, and it's super, super cheap, which is something that's really nice too. And what's also unique about it is it can measure the mechanical properties of single cell sheets, and also hopefully we can do double cell sheets. I think Nicholas you're doing those experiments right now, right? So what Dan did is he showed, again, coming back to this theme of the tire cord, so the tensile testing, except now these are with sheets of cells. And so you can actually look at them both along the direction and also perpendicular and measure their mechanical properties. And so what Dan did, this is a busy slide, but it's basically to show you that there is an anisotropy, which means there's a difference between in one direction or another direction. So they're 8 times stiffer in the direction of alignment. And this is significantly more anisotropic than prior studies. And part of this is no one had actually ever measured the mechanical properties of a single cell sheet before. And so what we're doing now with this basically is, so Tatiana Levins, she has developed this bioreactor, which again, this is quite interesting. You have different kinds of pulses between the age if you're 2 or 18. So our hope is to basically use this bioreactor to condition and use this to mimic a growing child. So then what we also did is we did some implantation of these cell sheets. This is what Josh Kim, another PhD student, has done. And so these are the different things we showed that we could use more than 5 different cell types using this cell sheet system. We could have the cells stay alive. And then, in fact, when you implant them, you can see that there's blood vessels that are growing towards them. So once we had this all done, the next step we wanted to do was to transition to translation, which is how do you then get this to the clinic? And so for that, we had to get the patents. So Li Tian, we work a lot with, and Mike Pratt. Another thing, you mentioned networking. So this tech, drugs, and rock and roll, which is a great networking point, that basically got us into thinking about how to translate our technology. I had one of my former postdocs, Elaine Lee, her former PhD advisor, Horst von Riekum, actually had started a couple companies. So he advised us to do the National Science Foundation I-Core program, Innovation Core program. And so we did that. This is myself, Josh, and this was our industrial mentor, Greg Martin, and then also talking a lot with David Friedman, who had gone through the I-Core program, and Jonathan Rosen, who actually implements a lot of these same kinds of ideas in his Masters of Engineering program. And then also I want to point out just the whole environment at BU, and these are the people that I've talked to or will be talking to, in terms of like, yeah, heads up, okay. No, so these are basically, sorry. So these are great, center directors and things that have great resources in terms of pushing forward translation. So we're really excited. And then I also have to thank John White for at first I was annoyed that you put me in a different class, right? But then I'm actually so excited teaching this class that was first started by Kathy Klaprich, BE 428, which is device design and diagnostics. Then also taken over by her postdoc, Jackie Linus, and then Mario Caboti and Allison Dennis. They just made it super easy for me to take over. And I've also been, not to take over, but to basically teach another section. And it's also been really informative to my own research in terms of like learning what the different design principles are. So what we've done is Josh and I essentially formed a company. This is Restore Biotechnology. So we graduated from the program, the NSF program, in March of 2015. So 2 years later, we started the company. And so as you learn, you have to pivot many times, but this is our minimal viable product. We're basically making a Band-Aid, a patch that will restore blood flow in blood vessels once they're damaged from, so you may be familiar with things like angioplasty or stenting. So that's the first story. The second story is a little bit newer. And so here we're interested in detecting and treating surgical adhesions, which are kind of I mean, people may have heard of this, but usually it's very underappreciated, but all the surgeons know that this is a problem. So it's a major cause of chronic abdominal and pelvic pain. It's a big cause of infertility in women. And then you have a lot, like for example, here you see a small bowel kink that's formed as a result of this adhesion. And so there are estimates of nearly $5 billion, and here this is the funding that we've received for this. And I just have to say, the inspiration for this is Art Stuckey, who really, so this is literally a text I get from him periodically. So this one, he just said, hey, Joyce, I met a guy, ortho guy at Baptist who said knee adhesions are his nightmare. So he'll just send me these to remind me, you know, like that adhesions are a problem. And so what our strategy is this Theranostics, right? So what happens in surgical adhesions is when you do that incision, you end up, your body has a normal response, it wants to heal, right? But instead of healing, it then can actually have two organs that are not normally wanting to touch each other. They touch each other, so they can form this, it's made of fibrin, basically fibrin. And so our idea is to put a vehicle in there that you can image it, and then basically you can use it to deliver drugs by releasing with ultrasound. So why do we pick ultrasound? A lot of you are probably familiar with ultrasound when you do ultrasound pregnancies. But we use it because it's cheap, but it actually does have some disadvantages in terms of it doesn't have that great of contrast, right? So that's why you need to use these microbubbles, which you can then expose them to an ultrasonic pulse, and then you'll see these bubbles. Oh, sorry, this one's not working. But basically, the bubble would then be expanding, and it would turn bright on the signal. So to just give you a sense of the length scale, these are the polymers, which are on the order of nanometers. But then here you have these microbubbles, which are on the order of 1 to 3 microns. And so the idea would be is that you can then see them brightly in this picture here. And so just as something I need to mention is this probably, you can't read, these are essentially the contrast agents that have been developed and are on the market. The main point is that all of these are used for perfusion. So in other words, you inject them, you can see them, they can tell you how fast something is moving, or if there's a blockage, but then they disappear. And you want them to disappear. But if you're trying to target something, you don't want it to disappear, right? And so especially if you're targeting maybe even cancer or cardiovascular disease, so it's a real problem. So what we decided to do is we have this strategy where we would use microfluidics to make them all the same size, and I'll tell you in a minute why that's important. Then we actually cross-link them so they can stay, so they actually won't be destroyed, right? Because what happens if you don't cross-link them, the bigger ones get bigger, and then the smaller ones get smaller, and then eventually the bigger ones pop and they disappear, right? And so our idea would be is that you can image with one frequency and then destroy them and release your payload with another frequency. So that's our general strategy. And so here's just showing you the reason why we wanted to make them all the same size is because that they have a different signal depending on their size. And so we use this microfluidic device where you can see they're basically being pumped out, and I can just speed it up to show you. You can collect these, and then you basically have your bubbles, and you can cross-link them using this chemistry, which is this diacetylene chemistry, where they actually turn color, too. They turn pink when you shine UV light on them. And you have this polymer, again, this theme of polymers. The polymers form on the shell. And so then you can actually see the pink ones float. The pink ones are the bubbles, and you see them floating. And so we also tested them. The main point for you to see here, this is a number fraction, is a function of time, is just to show you that our formulation, which are the top two here, they basically stay with time, whereas the other ones, the commercially available ones, disappear with time, as they were designed to, right? And here, just to show you, we can we estimate that we can last 10 hours, although my students say that they can last, several days up to a week. And so at this point, we actually had a problem because, so I don't know if you noticed this design of, and if anyone has done microfluidics, you'll agree with me that there's always a problem. You always get clogging. And so we actually did a calculation on this if we wanted to do preclinical studies in animals. We calculated that we would have to leave this device on for a week, nonstop, and basically to collect enough bubbles, right? And so the problem with that is that, as you know, you have to sit next to it because you don't want it to blow. Plus, you're putting this decafluorobutane in, which is this gas, right? And so we quickly said, okay, maybe this won't work. But fortunately, we had a separate collaboration with my collaborator at the ESPCI in France, Patrick Tobeling. And this is where the international part comes in again. So fortunately, they're experts in microfluidics. And so when I asked them if they could do, they basically have a multi-channel microfluidics. And so when I asked them, I said, yeah, for our single channel, it would take us, a week to make these bubbles. And he calculated, he did a little bit of calculation. He said, okay, maybe 100 seconds, right? So in other words, this is actually really exciting that this is just some of the bubbles that they showed us very, very recently. And the only reason why I put this up here too is, as you know, in France, they have like an important election next week, and it's really an issue. I think it's important to say here because science, someone was asking me recently, why is the March for Science, why are they marching in Berlin? Why are they marching in London? Why are they marching in Paris? And the reason is because science is global, right? You have collaborators. So this colleague of mine, he's a postdoc with Patrick, and so there was a scare a little bit. He had gone back to China and couldn't get back to Paris because there was a problem with his visa. And so these are the kinds of things I think it's really important that, we have to keep in mind and make sure that we don't go down some crazy rabbit hole. Okay, that's enough of okay. But basically, to show you, we can also target fibrin. We have a special peptide that we can target. And this shows you the numbers show that we have actually 6 times as much going to fibrin versus fibrinogen, which is the precursor. And just, I think this is my last kind of science slide here showing that, again, thanks to Art Stuckey, he developed this rat button model where they can put sutures and these little buttons into the peritoneum of a rat, and they can reproducibly form these adhesions. And that's really important because if you want to do platform testing of your theranostic vehicle, you need to be able to have a reliable model to be able to test these. So we're still kind of in the early stages of this in terms of like getting to our theranostics. And so those are kind of 2 examples of stories that I wanted to share with you. And then just to tell you that, again, this was an extremely collaborative project. Some of these were the very initial basic science studies or we were doing with bubbles and things. And actually something I forgot to mention, some of these flags, like some people have 2 flags too, And so this was a very highly kind of collaborative project. So finally, I just want to end with talking about, I mentioned women are very important too, and the women networks, starting of course with my mother. You already heard a little bit about her, but she also was always there when my children were much younger. They probably don't know this, but whenever I needed to like go travel or something, and maybe my husband and I, we were traveling at the same time, I'd say, Mom, can you come? And so she told me once, she said, me and my girlfriends, we call ourselves like the firewoman squad. When our daughters call, we come, and she said, and I expect you to do that too for Alicia. And I said, of course, Mom, And then also my sister, that's her there, that's me. And then my sister, and then Denise Denton, who was my freshman advisor at MIT, she was actually the first woman dean at a Research One university at University of Washington. All my wonderful friends at MIT undergrad that I still keep in touch with, we're the Langer Lab ladies here in grad school. And then I was really fortunate to have a wonderful group of postdoc women at Santa Barbara. Actually, all of them are professors. And Irene, who just keeps me in line. It's very difficult. I think I have the only picture of you, Irene. And then this is with arrows. has been really, really great. And Nancy for basically like introducing me to Qigong. That's been amazing. And then also outside of BU, this is the pictures, fun pictures at the latest AIMB meeting. And then also just my colleagues, Paula Raymond and Julie Chen, who've been amazing to work with, again, on these STEM equity, gender equity issues. And just as a quick plug, Julie's giving a talk next Thursday. Please all come to that. And I want to finally also acknowledge my grandmother, and this whole thing with flags, that's a Taiwanese flag. So I don't know if you guys know, so my grandmother, I always thought she was the same age as Taiwan, but I just found out recently she's actually a year older than Taiwan. So how many people can say that their grandmother's older than a country, right? She's 107. Those of you know that you actually are 1 year old when you're born, but she's still alive. She just came out of the hospital. She gave us a little scare, but she's great. This was actually taken from a FaceTime. It's so awesome that you can FaceTime with, anyway, so that was great, my ama. And then finally, I want to just thank my parents for giving me basically everything. And also, none of this would be worth anything without my family. TC, who's just always been there. I mean, I know you do all the cooking and everything, but it's like, but I do remember sometimes to cook. And then my wonderful daughter and son, Alicia, is actually getting an award tonight. She's getting a special award at the high school. So, but that's it. And I thank you for your attention, and we'll take any questions. So.
We have time for several questions, so think of some. Anyone?
Tyrone.
I was going to ask a question. Go ahead.
So the thermo-responsive scaffolds.
Yeah.
Do you coat them somehow with a protein or a collagen or something so that you do get absorption of the cells, or is it just treated almost like a fouling surface?
No, you can. I thought you were going to ask how you put the thermoresponsive polymer on, because I didn't talk about that. But basically, you can put, let's say, like fibronectin. You can. So that was actually one way I didn't mention. That's how you can stamp it on. But you can't. They will attach with just serum too. But yeah, and there's lots of different ways. Like the Okano group, they do electron beam deposition. We found, so Dan actually did a different method where he grafted them onto silicone rubber substrates, which we found to be, kind of a quicker way.
I have a question.
Okay.
Why are they aligned when you align the cell lines? It's much stiffer than when they're not aligned. The question is, why is it so much more aligned when you, and why is it so much stiffer, and can you control the stiffness to create the kind of mechanical property you want?
Well, you mean as opposed to if they're randomly oriented? Well, because you have the organization of, there's also extracellular matrix under there as well, right? And then one thing I didn't show you is they shrink quite a bit too when you pull them off. So I mean that's something so what's interesting is that the amount of collagen doesn't change between unpatterned and patterned, but it's just their organization. So you're pulling in that direction, right? But if you pull in this direction, it's going to be weaker, right?
Okay.
Yeah.
Can you control the stiffness to do what you want it to be?
You mean of the sheet?
Well, yeah.
I guess you could, if you could control, I guess, the how much protein is being secreted. What we have done in a different technique that Josh actually developed, we can actually change the stiffness of what the cells are sitting on, and then that actually controls what cell type the cell becomes. That's a different thing. Oh, what? Okay, Alice, what? Okay. Oh, Vic, okay.
Dr. Corey.
Very great. Seeing your whole history in the chart. Are those blood vessels going to remodel in in some way degrade and not aneurysmal dilatation is the bugaboo of all biographs that I've ever seen.
Yeah, so what he's talking about is about, I guess 20 or 30 years ago, there was a lot of work done with basically collagen fibers where people were seeding cells on them and they would implant them into animals. And the problem is that they would aneurysmally dilate, right? So it's a kind of like a balloon and it would just balloon out. And so we would obviously have to see like if there's elastin in there. So we haven't done long enough term tests to see if there be aneurysm dilation or not. But in the minimally viable product that we're trying, that's just going to be a surface coating. So we're hoping that will actually be in the inner layer of the vessel. So we're hoping that won't be an issue. But you're absolutely right. It's a big problem. And so those of you who know, you might have to have elastin in there, which is, I can show you. So this is collagen, and then if it bounces back, good. Okay. So that means you have elastin. So, okay. Okay, Andre? Yeah.
On your opening slide, you had an image of a blood vessel that looked remarkably similar to a coax or fiber optic. Do you think there's any connection there?
Well, I mean, there's certainly a lot of people using optical fibers in, like OCT for imaging of vessels, certainly.
No, what I meant
you mean to use that structure to make a fiber?
I was just wondering about the coincidence that the blood vessel looked the structure looked very, very similar to a coax.
You mean
or
it has
fiber optic.
the different helical?
The different layers and.
Okay, yeah, no, I wasn't aware. I mean, I know about the optical fibers, but I didn't know that there was I just thought they're all collected. I didn't realize so maybe we should talk, yeah.
There's another question.
Vicki, did you have?
That was a great talk, Joyce. Do your patches resolve the problem of calcification since that prompted the beginning?
Yeah, so we haven't done long enough tests. I guess the best animal model for that is a sheep. And interestingly enough, I have another student who's actually looking at trying to detect calcification early with, with as you know, we work with you on the superparamagnetic, the iron oxide nanoparticles. So we're trying to see if we can detect valve calcification early. So we haven't done long enough studies where calcification would occur. But like I say, the sheep is a great model because it has accelerated calcification in the sheep for whatever reason, I don't know.
Question? Only, can I give Joyce something? Hold on.
Oh. It's a what?
It's a guitar. First,
Oh.
let's thank Joyce again for a wonderful talk. And we have an endowed physical chair for you. It's engraved.
Oh.
That's your name. You do the same name to everybody. No. The children get to carry you back in the rain, back to your. We'll get it back to the office for you.
Okay.
Okay, we have a wonderful reception outside for everyone. Thank you for coming. Thanks again to Charles for making this all possible. And let's socialize.